I am a pharmacist with a degree in public health who also happens to have neuropathy. I recently began a new job as a pharmacogenomics testing representative, which further educated me about genetics and medical genomics. I am also a board member of the Western Neuropathy Association. As your board, we decided that we wanted to present a webinar that would remind our members that we can still connect with our family members at the holidays, even during COVID. Therefore, today I will be talking about genetics and family history, a unique way to bridge the distance between us during the holidays. I will try to break down the basics of genetics, discuss why it is an important part of our family history, and present an example of medical genetics in neuropathy today. There may be times when you will find yourself wondering if you are back in biology or chemistry class. So many terms, so many pronunciations. Never fear, there will be time for questions at the end. Most of us know that we can reduce our risk of disease by eating a healthy diet, getting enough exercise, and not smoking. But did you know that your family history may be one of the strongest influences on your risk of developing heart disease, stroke, diabetes, or cancer? Even though you cannot change your genetic makeup, knowing your family history can help you reduce your risk of developing health problems. Family members share their genes as well as their environment, lifestyles, and habits. Everyone can recognize traits such as curly hair, dimples, leanness, or athletic ability that run in their families. Risks for diseases such as asthma, diabetes, cancer, and heart disease also run in families. Everyone's family history of disease is different. The key features of a family history that may increase risk are diseases that occur at an earlier age than expected, 10 to 20 years before most people get the disease. Disease in one or more close relative. The disease that does not usually affect a certain gender, for example, breast cancer in a male. Certain combinations of diseases within a family, for example, breast and ovarian cancer, or heart disease and diabetes. If your family has one or more of these features, your family history may hold important clues about your risk for disease. People with a family history of disease may have the most to gain from lifestyle changes and screening tests. You cannot change your genes, but you can change unhealthy behaviors such as smoking, inactivity, and poor eating habits. In many cases, adopting a healthier lifestyle can reduce your risk for diseases that run in your family. Screening tests such as mammograms and colorectal cancer screening can detect diseases like cancer at an early stage when they are most treatable. Screening tests can also detect disease risk factors like high cholesterol and high blood pressure, which can be treated to reduce the chances of getting a disease. Genetics basics, 6 things you should know. Human genetics is a branch of biology that studies how human traits are determined and passed down among generations. Let's explore this exciting field to better understand how your genes help shape your traits and health. For genetics basics, there are 6 things you should know. DNA, what it is, and what does it do. First, we'll learn about DNA, chromosomes, and genes, the building blocks of the genetic code, and how they result in human traits. Diversity and variation among humans. Next, we'll learn how variation and diversity are important in human genetics research and its health implications. Inheritance, health, and disease. Talking about inheritance, health, and disease, we'll discover how genetic traits are passed down through families and how they may affect health. Family history. By collecting your family's health history, you can learn what health problems you may be at increased risk for in the future and how to reduce your risks. Genetic testing, privacy, and healthcare. Next, we will seek to understand genetic testing options, privacy protections for your genetic information, and how you may encounter genetic testing in healthcare. And medical genetics. Finally, we will develop an understanding of how variations in an individual's DNA may affect disease and health, which is the focus of genomic medicine. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, where it is called nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA. Mitochondria are structures within cells that convert the energy from food into a form that cells can use. DNA, the body's instruction manual. Humans and all other living things have DNA information. The information in your DNA gives your cells instructions for producing proteins. Proteins drive important body functions like digesting food, building cells, and moving your muscles. Your DNA is the most unique and identifying factor about you. It helps determine what color your eyes are, how tall you are, and how likely you are to have certain health problems. Even so, over 99% of DNA sequences are the same among all people. It is the remaining 1% that explains much of what makes you, you. DNA is arranged like two intertwined ropes in a structure called a double helix. Each strand of DNA is made of 4 types of molecules, also called bases, attached to a sugar phosphate backbone. The 4 bases are adenine, A, guanine, G, cytosine, C, and thymine, T. The bases pair in a specific way across the 2 strands of the helix. Adenine pairs with thymine, and cytosine pairs with guanine. So what is the difference between DNA and RNA? DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are very similar molecules that serve very different functions. DNA is responsible for long-term storage of the genetic code. It resides in the nucleus of cells. RNA, on the other hand, is responsible for several important processes, including transporting the genetic information found in the DNA to other sites in the cell where it can be used to make proteins. It is found in the cytoplasm of a cell. Despite their different jobs, DNA and RNA have almost the same basic structure. Each one is made up of nucleotides. These tiny subunits are made of a sugar, a nitrogen base, and a phosphate group. The differences start to show up when we take a closer look at their structures. The most obvious difference is that DNA is a double-stranded molecule, while RNA is a single-stranded. DNA is also much longer than RNA. An entire chromosome is actually just one molecule of DNA. While both DNA and RNA have sugar molecules in their subunits, those sugars are slightly different. DNA uses deoxyribose, but RNA uses ribose, which has an extra hydroxyl OH group tacked on. DNA and RNA also have nearly identical nitrogenous bases. Both have the bases adenine, cytosine, and guanine. However, DNA uses a 4th base called thymine. RNA's 4th base is uracil. The only difference here is that uracil is missing a methyl group, CH3. Chromosomes, packages of DNA. Genes are packaged into tightly wound lengths of DNA called chromosomes. Humans have 23 pairs of chromosomes. Sex chromosomes, identified as X or Y, determine whether a person is male or female. Each chromosome can be identified by its size and shape under a microscope. Each has a specific set of genes that is the same from person to person. One copy of each chromosome in a pair is inherited from each parent, which means that you inherit one copy of each gene from your mother and one copy from your father. Each chromosome has a centromere at its center, which is a small structure that divides the chromosomes into 2 parts. Each part is called an arm. Genes are located on the arms of chromosomes. Genes. Coding for proteins and traits. Genes are small segments of DNA that have different functions. Many, but not all genes, make the proteins that our bodies need to function. You have 2 copies of each gene, one on each chromosome in a pair. Genes that code for proteins come in different versions called alleles. Alleles of a gene have differences in the precise DNA sequence. A common example of this is eye color. We each have the same genes for eye color, but a different allele combination within those genes results in different eye colors. DNA is the genetic blueprint that codes for and determines the characteristics of an organism. Traits are your observable characteristics. Many physical traits are genetic. Genetic differences give our bodies information that result in traits that differ from person to person. We can also use genetic information to determine what inherited traits you may have. A difference from an expected sequence of a gene is called a variant or mutation. Variants can be inherited from your parents, or they can happen spontaneously. All of us have variants, but not all variants are harmful. Variants that are harmful can cause or increase our risk for certain diseases. Over 99% of all DNA sequences are identical among all people. The small percentage that is different helps make each person unique. Those small differences explain why some people have blue eyes and others have brown eyes. They explain why some people are colorblind, while others can differentiate between turquoise and cerulean. Variation and diversity are important when it comes to research. In genetics, an important research goal is to improve human health. To develop treatments that will be effective in all patients, research must include people from a range of backgrounds. Increasing diversity in human genetics research will help to ensure that everyone benefits from discoveries. Inheritance, health, and disease. Genetic diseases are caused by random changes in our DNA called variants or mutations. When a variant occurs, it can change the way a gene functions or the protein it codes for. This change can cause a disruption in the body that may cause a disease. These changes can be passed on to the next generation in different ways. Autosomal dominant diseases are caused by variants in genes found on non-sex chromosomes. For these diseases, only one altered copy of the gene in each cell is needed for a person to be affected. In most cases, an affected person inherits the condition from a parent who has the disease. Huntington's disease is inherited via autosomal dominant genetic change. A person who has one change in the HTT gene will develop Huntington's disease, even if the second copy of the gene is healthy. Autosomal recessive disease genes are also found on non-sex chromosomes. For a person to have an autosomal recessive disease, both copies of the gene must be altered. Many diseases, such as cystic fibrosis, follow an autosomal recessive inheritance pattern. Carriers are people who have only one altered copy of a gene for a recessive disease. Since 2 altered copies are necessary to have the disease, a carrier would not have the disease, but would carry it on his or her DNA. Carriers of an autosomal recessive disease are at risk of having children with the disease, but only if their partner is also a carrier of the same disease. X-linked recessive diseases are caused by genetic variants on the X chromosome. In males who have only one X chromosome, only one altered copy of the gene is necessary to cause the disease. Because females have 2 X chromosomes, they are less likely to have an X-linked disease. If you do inherit an X-linked disease, they may be less severely affected. Many females who have only one altered copy of the gene are unaffected carriers. Red-green color blindness is an example of an X-linked recessive disorder. X-linked dominant diseases are caused by mutations in the X chromosomes. Only one altered copy of the gene is needed to cause the disease. Neuropathies are nervous system disorders that cause nerve damage. They affect the peripheral nerves, including nerves beyond the brain and spinal cord. Hereditary neuropathies are passed on genetically from parent to child. They're sometimes called inherited neuropathies. Neuropathies can also be non-hereditary or acquired. Acquired neuropathies are caused by other conditions such as diabetes, thyroid disease, or alcohol use disorder. Idiopathic neuropathies have no apparent cause. Hereditary and non-hereditary neuropathies have similar symptoms. Symptoms of hereditary neuropathy depend on the group of nerves affected. They can affect the motor, sensory, and autonomic nerves. Sometimes they affect more than one nerve group. For instance, Charcot-Marie-Tooth, abbreviated CMT disease, one of the most common types of hereditary neuropathies, affects the motor and sensory nerves. Hereditary neuropathies can have similar symptoms. Some of the most common symptoms are sensory symptoms, which include pain, tingling, or numbness, often in the hands and feet. Motor symptoms, which include muscle weakness and loss of mass, otherwise known as muscle atrophy, often in the leg, feet, and lower legs. Autonomic symptoms, which include impaired sweating or low blood pressure after standing up from sitting or lying down. And physical deformities, which include high foot arches, hammer-shaped toes, or curved spine, known as scoliosis. Hereditary neuropathy symptoms can range in intensity from mild to severe. In some cases, symptoms are so mild that the disorder goes undiagnosed and untreated for a long time. Symptoms don't always appear at birth or during childhood. They can appear during middle age or even later in life. There are many types of hereditary neuropathies. Sometimes neuropathy is the distinguishing feature of the disease. This is the case with CMT. In other cases, neuropathy is part of a more widespread disorder. More than 30 genes have been linked to hereditary neuropathies. Some genes haven't yet been identified. The most common types of hereditary neuropathy are as follows. Charcot-Marie-Tooth, abbreviated CMT disease. CMT disease refers to a group of hereditary neuropathies that affect the motor and sensory nerves. Approximately 1 out of 3,300 people is affected by CMT. There are many genetic subtypes of CMT. CMT type 1A, abbreviated CMT1A, is the most common. It affects approximately 20% of people who seek medical treatment for symptoms caused by an undiagnosed peripheral neuropathy. Symptoms of CMT depend on the genetic subtype. The disorder can cause many of the symptoms listed above. Other symptoms can include difficulty lifting the foot or holding it horizontally, unsteady gait or balance, and poor hand coordination. At least 4 genes are involved in the transmission of CMT from parent to child. Children who have a parent with CMT have a 50% chance of inheriting the disease. A child can also develop CMT if both parents have recessive copies of the abnormal gene. And hereditary neuropathy with pressure palsies, abbreviated HNPP. People who have HNPP are extremely sensitive to pressure. They might have difficulty carrying a heavy shoulder bag, leaning on an elbow, or sitting on a chair. This pressure causes episodes of tingling, numbness, and loss of sensation in an affected area. Commonly affected areas include the hands, arms, feet, and legs. These episodes can last for up to several months. Over time, repeated episodes can lead to permanent nerve damage and symptoms, such as muscle weakness and loss of sensation. People with HNPP may experience chronic pain, especially in the hands. An estimated 2 to 5 out of 100,000 people are believed to be affected by HNPP. A child born to a parent with HNPP has a 50% chance of developing HNPP. Having a family member, and especially a parent, who's been diagnosed with hereditary neuropathy is the most significant risk factor. Some studies suggest that health conditions such as type 2 diabetes and obesity may increase your risk for certain hereditary neuropathies. More research needs to be done to understand the link between health conditions and hereditary neuropathies. Your doctor may refer you to a nerve specialist known as a neurologist to help diagnose your condition. You may need several tests before your doctor can reach a hereditary neuropathy diagnosis, including genetic testing. Genetic testing can be used to identify genetic abnormalities linked to hereditary neuropathies. Biopsies. A biopsy is a minimally invasive procedure that involves taking a tissue sample and looking at it under a microscope. This test can help identify nerve damage. Nerve conduction tests. Electromyography is used to help doctors understand the nerve's ability to carry an electric signal. They can help identify neuropathies. Nerve conduction tests can help identify the presence of a neuropathy, but they can't be used to determine whether the neuropathy is hereditary or acquired. Neurological evaluations. These tests assess your reflexes, strength, posture, coordination, and muscle tone, as well as your ability to feel sensations. Additional tests may be used to rule out other health conditions or identify injuries related to the neuropathy. Hereditary neuropathy can be diagnosed at any age. However, symptoms for certain types are more likely to appear during infancy, childhood, or early adulthood. In many cases, there is no cure for hereditary neuropathy. Instead, you'll need ongoing treatment to manage your symptoms. Common treatments include pain medication, physical therapy, corrective surgery, and therapeutic shoes, braces, and supports. However, for some hereditary neuropathies, breakthroughs in medical genomics has brought new hope for neuropathy sufferers. Eating a balanced diet and getting regular exercise is also recommended. If you have a loss of sensation, your doctor might suggest certain safety measures to help you avoid hurting yourselves. Hereditary neuropathies can't be prevented. Genetic counseling is available to parents who might be at risk of having a child with an inherited neuropathy. If you're experiencing symptoms of a hereditary neuropathy, you should make an appointment with your doctor right away. Early diagnosis can improve the long-term outlook. Keep a record of your symptoms to show to your doctor. If possible, identify whether neuropathy affects other members of your family. The long-term outlook for people who have been diagnosed with hereditary neuropathy depends on the genes affected, as well as the type of neuropathy. Certain types of neuropathy progress more quickly than others. In addition, hereditary neuropathy symptoms can be mild enough to go undiagnosed for a long time. Symptoms can also be severe and disabling. If you have hereditary neuropathy, talk to your doctor about what to expect long term. Your family history holds key information about your past and clues to your future health. Many of your physical traits, such as eye color, hair color, and height, are inherited. So too are risks for certain genetic conditions and health problems, such as heart disease, diabetes, and some cancers. You may have noticed that some of your relatives are healthier and live longer than other relatives. You may also have noticed that some relatives have the same health problems. By collecting your family's health history, you can learn what health problems you may be at increased risk for in the future and how to reduce your risks. For instance, people at increased risk for heart disease may be able to reduce their risk through not smoking, regular exercise, and diet. Learning your family history can benefit both you and your relatives, and it can also bond you together. When members of a family can pass traits from one generation to another through genes, that process is called heredity. Family members share genes, behaviors, lifestyles, and environments that can influence their health and their risk for diseases. Hereditary diseases can be higher in some families than in others, and your chances of having a hereditary disease can go up or down depending on your age, sex, and race or ethnicity. Genetic factors likely play some role in high blood pressure, stroke, and other conditions such as hereditary neuropathy. People with a family history of neuropathy are also likely to share common environments and other potential factors that increase their risk. The chances for hereditary neuropathy can increase even more when heredity combines with unhealthy lifestyle choices, such as alcohol abuse and eating an unhealthy diet. You can find out more about genetics and disease on the CDC's Office of Public Health Genomics website. Family health history is a record of the diseases and health conditions that happen in your family. Family health history is a useful tool for understanding health risks and preventing health problems. To help people collect and organize their family health information, the CDC's Office of Public Health Genomics Unit worked with the U.S. Surgeon General and other federal agencies to develop a web-based tool called My Family Health Portrait. You should check it out. Genetic testing, privacy, and healthcare. There are many different tests available for genetic diseases. Some people use direct-to-consumer testing to see whether they are at risk for certain genetic diseases. Genetic testing is also available through a genetic specialist or your primary care physician for things such as cancer screening, cardio screening, and pharmaceutical diagnostic tools. In the medical setting, your doctor will first determine if you would be a good candidate for genetic testing. People who have relatives with a genetic disease are often directed to genetic testing. There is also a counseling component, which explores what you plan to do with the results and how the results may affect you and your health. Remember, a positive genetic test result does not necessarily mean that you have the disease now or will have it in the future. For many diseases, a positive genetic test can simply represent a higher than average risk of disease. This is especially true when looking at diseases that have environmental causes or gene-environment interactions. Your doctor may offer you genetic testing under certain circumstances. You could receive a genetic test because you're pregnant and want to learn more about your baby, because you have a family history of a genetic disease, or to confirm a suspicion that you have a genetic disease. Some people receive genetic testing before or during pregnancy to see if they carry certain genetic variants that can be passed down to their children and cause specific diseases. People who carry the variants but do not have the disease are called carriers. They do not have the genetic disease, but if two carriers have a child together, the child could inherit the disease. You can take direct-to-consumer genetic tests without involving your healthcare provider. Many testing companies require you to mail in a DNA sample. After analyzing the DNA, they will send you the results directly. It is important to consider potential effects of testing and the privacy policies of the companies offering the test. Ancestry testing is a way to find out more about your family of origin and its history. It can be done without involving your healthcare provider and is especially informative for people who do not know much about their biological family. Common ancestry tests are 23andMe, Crygenetics, MyHeritage, Ancestry DNA, Living DNA, and Family Tree DNA. There is even genetic testing for dogs, such as Wisdom Panel. The Genetic Information Non-Discrimination Act, abbreviated GINA, is a federal law that protects against genetic discrimination in the workplace and health insurance. It also safeguards individual and family privacy of genetic information. With new advances in science, technology, and data management, GINA plays a vital role in establishing a baseline of protection for all Americans against discrimination. Most healthcare providers are considered covered entities and must comply with the HIPAA privacy rules, with regards to protected health information, including genetic information. More information about HIPAA can be found at hhs.gov. More research information Most research information is anonymous and only seen by those completing the research. When considering participating in a research study, be sure to ask how they will protect your genetic information. It is estimated that 3 to 7% of the general population will be diagnosed with a recognized genetic disorder. This does not include common disorders such as cancer, diabetes, heart disease, and psychiatric disorders. All diseases or medical conditions have a genetic component, except trauma. Understanding how variations in an individual's DNA may affect disease and health is the focus of genomic medicine. This knowledge can lead to innovative ways to diagnose disease, early detection by identifying genetic predisposition to a particular disease, new approaches to treatment, and development of designer drugs that target a mutation or its products, called pharmacogenomics. Common terms in medical genetics are genetics, which is the study of genes and their effects. Genomics is a study not of single genes, but of the functions and interactions of many genes in the genome, which is an organism's complete set of DNA, including all of its genes. Medical genetics is an application of genetic principles to medical practice. This includes studies of inheritance, mapping disease genes, diagnosis and treatment, and genetic counseling. Some of the top genetic companies are Admira Health, Teloyears, Origin, and Helix. Pharmacogenetics is the study of how drugs affect the body with respect to specific genetic backgrounds. Knowledge of these effects can improve effectiveness of drugs and minimize side effects on an individual patient basis. Many companies such as Genomind and GeneSight test for psychiatric medications. With a new innovation called next generation sequencing, PGX1 Plus by Admira Health has emerged, which tests 18 different therapeutic categories over 300 medications. Pharmacogenetics testing is a relatively new tool in your doctor's tool belt for picking the right drug at the right time in the right dose. Over 80 medications now require pharmacogenomics testing prior to dispensing, including cancer drugs, opiates, and psych drugs. Testing is simple with an oral swab, which is sent to the lab for DNA sequencing. The DNA results provide a lifetime utilization of information about your DNA involved in drug metabolism, potential interactions with drugs such as anesthesia called malignant hypothermia, a deadly reaction encountered on the operating table, and predispositions to hereditary diseases such as addiction, among other health information. Pharmacogenomics testing is covered by Medicare and other major health plans because of its beneficial information in aiding with optimal medication prescribing and general health prediction and care. Amyloidosis is a protein disorder. In this disease, proteins change shape or misfold, then bind together and form amyloid fibrils, which deposit in organs. As amyloid fibrils build up, the tissues and organs may not work as well as they should. Our bodies make several proteins that can cause amyloidosis. To choose the right treatment, it is very important to know the exact protein that is causing the disease. The two most common types are light chain, abbreviated AL, and transthyretin, abbreviated ATTR, amyloidosis. Transthyretin, also called TTR, is a protein made by the liver that helps carry thyroid hormone and vitamin A in the blood. Normally, TTR is made up of 4 identical parts. However, in ATTR amyloidosis, the protein becomes unstable, breaks apart, and deposits in the heart and or nerves. There are 2 types of ATTR amyloidosis: hereditary or familial ATTR amyloidosis. In this form, there is a change or mutation in the DNA that is inherited and can be passed from one generation to the next. This makes the TTR protein more unstable, and more likely to form amyloid fibrils. Different mutations lead to different symptoms. Some may affect the nerves, some may affect the heart, and some may affect both. Wild-type ATTR amyloidosis, unlike hereditary ATTR amyloidosis, wild-type does not involve abnormal DNA and cannot be passed on to family members. Instead, as you get older, the normal TTR protein becomes unstable, misfolds, and forms amyloid fibrils. These ATTR amyloid fibrils usually deposit in the wrist in a narrow pathway called the carpal tunnel. They can cause carpal tunnel syndrome, which causes your hand and arm to become numb and tingle. The spinal canal, which can cause narrowing of the spinal column, called spinal stenosis. The heart, where it can cause heart failure and or irregular heart rhythm, called atrial fibrillation. Carpal tunnel syndrome and spinal stenosis can develop several years before heart failure. Early diagnosis of familial ATTR amyloidosis is very important to successful treatment. Because the disease is inherited, genetic screening can be an effective way to identify a patient's family members who may carry the mutation. If an individual is identified as carrying the genetic mutation for familial ATTR amyloidosis, further screening for manifestations of the disease is required. Additional screening will continue on a regular basis. Genetic counseling and testing for familial ATTR amyloidosis is available through the Stanford Amyloid Center and other research hospitals around the country. Amyloidosis can affect all of these areas in the human body. Symptoms depend on the organs that are involved. Symptoms indicating the arms are affected include carpal tunnel syndrome, numbness and burning and or tingling, otherwise known as peripheral neuropathy, and biceps tendon rupture. Symptoms indicating the back is affected include lumbar spinal stenosis. Symptoms indicating the legs are affected include swelling of the feet or legs, numbness, burning and or tingling, again, peripheral neuropathy, and leg weakness. Symptoms indicating the head and neck are affected include eye floaters or vitreous opacities and lightheadedness upon standing. Symptoms indicating the heart and lungs are affected include shortness of breath, palpitations, chest pain, and fatigue. Symptoms indicating the stomach or intestines are affected include poor appetite, bloating or excess gas, and diarrhea or constipation. Several medications have been approved by the Federal Drug Administration, abbreviated FDA, for treating patients with ATTR amyloidosis. Other medications continue to be investigated. ATT silencers. These medications act on the liver to decrease the production of TTR. 2 ATTR silencers have been approved by the FDA to treat patients with the hereditary type of ATTR who also have neuropathy. Patrisan, otherwise known as Onpatro, is an infusion that is given every 3 weeks. Patrisan, like many investigational drugs, including our vaccine for COVID, uses RNA interference to achieve its therapeutic effects. Inotersen, otherwise known as Tegsedi, is an injection given once a week and requires weekly lab work. Antisense oligonucleotide drugs, abbreviated AOs, are short strands of oligonucleotides that bind to RNA and prevent translation. The preliminary data from an open-label phase 2 trial of ISIS-TTRRX, which is an investigational drug, revealed that 22 heart disease patients with wild-type and mutated ATTR who received weekly injections achieved a positive therapeutic effect with a favorable safety profile. ATTR stabilizers. These medications stabilize the TTR protein, which in turn prevents it from breaking apart and forming amyloid fibrils. Tafamidis, otherwise known as Vyndamax or Vyndaqel, is approved by the FDA for patients with hereditary or wild-type ATTR that affects their heart. Tolcapone binds to the thyroxine binding pocket at the TTR interface, making it a stronger aggregation inhibitor than tafamidis. AG10, made by iDose right here in Northern California, is a medication currently being tested in a clinical trial. AG10 was designed to mimic naturally occurring variant of the TTR gene, abbreviated T119M, which also binds to the thyroxine binding pocket at the TTR interface. And it is considered a rescue mutation because it has been shown to prevent ATTR in individuals carrying disease-causing mutations in the TTR gene. To our knowledge, AG10 is the only TTR stabilizer in development that has also been observed to mimic the super-stabilizing properties of this rescue mutation. Diflunisal, otherwise known as Dolobit, is a non-steroidal anti-inflammatory drug, abbreviated NSAID, that has been shown to also stabilize the TTR protein. However, this medication has not been fully studied in patients with ATTR that has affected the heart and also may not be tolerated well due to side effects. And last, there are fibril disruptors. These medications may help break up and clear ATTR amyloid fibrils. Doxycycline, which is an antibiotic, and green tea extract, an over-the-counter supplement, have only been tested in small studies, and there is limited evidence that these medications would be helpful in treating amyloidosis. Prothena, another biopharmaceutical company in the Bay Area, is diligently working on PRX004, an investigational monoclonal antibody designed to specifically target and clear the misfolded and toxic forms of the TTR amyloid protein found in ATTR amyloidosis. Because TTR is made in the liver, a liver transplant is also a possible treatment for patients with some hereditary forms of ATTR amyloidosis, but not for those with the wild type form. So you can see that alongside the biopharmaceutical companies that are working tirelessly on a vaccine for COVID-19 virus, there are other biopharmaceutical companies who are passionate about bringing new hope to people with hereditary neuropathies through medical genomics. So whether you are inspired to find out more about your family heritage or want to enhance your healthcare through medical genomics testing, realize that genetics offers an endless vision into what makes you you. Feel free to research my references and more about genetics, hereditary neuropathies, and advances in medical genomics. And know that as this unique holiday season is upon us, whether you are joining with relatives to find your common bonds or discovering your own unique characteristics, we are all in this together. Thank you very much.